Okay, it seems that probably some of your classmates won't be present this afternoon, but nonetheless, let's proceed with our seventh exercise. So I hope you had a wonderful reading break. So I hope you're well rested, you're already energetic, and I hope the reading break had brought you a lot of inner peace of mind. Now, for today, we will be doing the laboratory exercise number 7, and we will have a guest lecturer for this afternoon. Now, before our guest lecturer would start his presentation later, around 1:30, I would like to discuss some of the learning module for this exercise. So the learning outcome for this exercise is the title rather of the exercise is breeding crops with resistance to pests and diseases. So it's breeding for biotic stresses, particularly for pests and diseases. And not just any disease, but disease caused by your pathogens and, of course, living organisms in general. At the end of the exercise, the student should be able to identify the different sources of resistance and breeding methods to incorporate them. So at the end of the exercise, you should know which plants to use or what could be the possible sources of resistance for your plant and how difficult it is to incorporate the resistance to your desired genotype, and the breeding methods in which you try to incorporate them, the difficulty depending on the source of resistance as well. You may also describe the general methods of screening methods of disease resistance. This will be discussed by our guest lecturer for this afternoon, which he will be discussing the general methods and practices in biotics resistance screening. So try to look out and watch out for it and ask questions if you'd like. So he's a very well-known researcher from the University of the Philippines, Los Baños, in the Institute of Plant Breeding. And of course, at the last part is develop a breeder's toolbox for resistance breeding for your favorite pathogen. So what I would like you to do here is to accumulate some literary sources through the worksheets in which it in which these sources or information could help you decide which breeding method to do, or what could be the necessary state of art or the presence of certain information of the particular pest that would help you in your breeding for biotic stress, or your breeding goal, which is breeding crops with resistance to pests and diseases. Okay, so the learning content is quite easy. So first we discuss the importance of crop diseases in the first paragraph, and then we discuss in the second paragraph the different types of organisms that incite diseases, biotic stresses, which could be your as could be vertebrate pests, and even as small as viroids, but could also be in between, like your phytoplasmas, your vascular wilts, and even your plant parasites or your parasitic plants. Then, after which, we discuss the different strategies how plants cope up with this disease. So it could be through avoidance, resistance, or tolerance. Although, since our lecture will only focus on resistance, we will define only resistance in this module. And since resistance is the one commonly used as a breeding strategy in order to serve as a defensive mechanism in order to incorporate higher yields in a place where there is high density of pests. Also, there are different mechanisms and types of resistance that will be discussed. It's already summarized here. So I've taken the liberty to summarize the sources of resistance for you. So it's a broad resistance, non-host resistance, type of host resistance, the hypersensitivity, and the partial resistance. So in here, you will be discussing what type of resistance there is or are available, and could it be possibly monogenic or oligogenic, or even using or could it be quantitatively inherited by more than one genes? So we will be discussing our genes here, and of course, the gene for gene model is also described. Okay, and as an example is placed here also in the summary. Okay, so just how to go about this module. Then, after which, we also describe the various sources of resistance. As discussed, you need to identify the sources of resistance and how to incorporate them as an objective, right? So it could be the cultivar, the commercial cultivar, land races, and wild progenitor, their descriptions, the presence of desirable traits, and of course, the difficulty of incorporating this resistance to the desired genotype. So this is a summary table as well that is made for you so that you could easily understand. This course is actually a whole 3-credit course in in Wageningen University. So I just tried to at least introduce some of the very salient features that you might be needing in your search for the breeding for resistance. Okay, so aside from this description, we also discuss that depending on the type of pest, you will also determine which type of mechanism of resistance would you employ. So here is a strategy, dichotomous scheme, more likely a question and answer type of strategy, in which a diagram that would help you assist the breeding strategy necessitated depending on the evolutionary risks posed by the pathogen, because sometimes your pathogens might evolve, and therefore with the evolution of that pathogen, depending on how fast a pathogen or a certain pest evolves, then there might be also higher amounts of your hybrids might easily break down, okay, or your plants that are resistant might easily break down. So in here it takes into account this strategy. And also, aside from that, we discussed the various breeding methods. So there are a lot of breeding methods, and all breeding methods discussed in exercise 5 and 6 could be actually employed for resistance breeding. So there's nothing special to it, but there are some breeding procedures that are most likely used for incorporation of resistance, and we outlined it here, including backcrossing, recurrent selection, and of course your marker assisted breeding. Okay, so then after which we also discuss that of course many pests pose evolutionary risk, as I've said, and uh, the higher the evolutionary risk, the greater chance of breaking down the resistance gene, and this is given by and it is just a function of the two important parameters according to the model of MacDonald and Linde in 2002, which is the gene flow. Higher the gene flow, the higher the evolutionary risk, and of course the reproduction or mating system. The more allogamous it becomes, or if it becomes more mixed, then the higher the evolutionary risk. Okay, so and both of the two are could be actually be correlated, or can be can increase the evolutionary risk of a particular gene, resulting to the breakdown of resistance genes. Okay, so we ended up there, but I also made available in the Google Classroom a supplementary material, this paper by MacDonald and Linde, if you want to read more on this. Okay, so the learning activity for this afternoon is that you will have a virtual introduction. In this afternoon, we will be choosing the Institute of Plant Breeding breeder and breeding programs and resistance screening methods. So each student is expected to submit a report that summarizes the knowledge they've gained from the virtual visit and realizations they have made regarding the breeding and screening methods for resistance in general. So it's the same for your narrative reports in exercise 5 and 6. But more or less, I would just like you to summarize the salient features and make some reflections of what you've learned from the speaker or our guest speaker this afternoon. So, and then in activity 7.2, you'll create a breeder's toolbox, which is a set of questions that would require you to develop to answer. Okay, and in which could help you eventually in making a decision process in which breeding method to choose, how to approach basically the breeding for abiotics for biotic stress. Okay, by answering worksheet 7.1 and of course the study questions. Now the required output as summarized would be number one, the accomplished worksheet 7.1, answers to the guide questions, and of course your narrative report. The breakdown, this is not 6.1, kindly disregard. This is 7.1, is equivalent to 56 points. Study questions is 14 points, and the narrative report is 30 points. 56 points because each cell inside the worksheet, which you would find, is around 3 points, and the reference would be 1 point. Okay, and study questions is 5 points for the 2 questions and 4 points for 1, and narrative report for a total of 30 points. So the component for the narrative report is just this one. So just multiply. So if you want to have an outstanding narrative report, make sure you have a complete information and supporting details with reflection. Okay, so that's just very basic. So the references are as follows. This is heavily referred to in a book by Nix, and Dr. Yuling Bai, one of our professors in the Netherlands, in breeding for crops with resistance to disease and pests. Now, also it's also heavy on the McDonald and Linde paper in the population genetics for plant pathogens and resistance. So formerly, this exercise in a face-to-face setting would only require you to find out the screening methods. But since the course is actually breeding methods, so we must relate the breeding methods to the objective, which is for biotic stress resistance. Okay, so here is the worksheet 7.1, which is some of the question, which is a total of 56 points. So what is the name of the pathogen? You need to answer. A species name. This is a species name. Kindly kindly correct if ever. What is the reproduction or mating system of the pathogen? So there are questions on the reproduction or mating system of the pathogen. So, and aside from that, you also known if it is sexual, asexual, or is it mixed for the mating system? Is it spreading soil or airborne? So it depends, because this would help you identify by Linde in their functionality whether it's evolutionarily much more evolutionary risky. Okay, so and based on gene flow. Symptoms of the disease incited by the pathogen is the genome sequence, is the pathogen's genome sequence. Other notable characteristics of the pathogen. In here, you can place probably some notable characteristics that it brought a particular particular economical damage in this time of the year or this time of the season, or so on and so forth. So any significant or salient feature may be placed in this question. Other notable characters, what type of lifestyle of the pathogen? Is it biotrophic, necrotrophic, and hemibiotrophic? So in this one, you need to first try to answer the question, laboratory's guide question, so that you would know what biotrophic, necrotrophic, and of course hemibiotrophic mean, which is just the pathogen lifestyle. Does your favorite pathogen have high genetic diversity? And if some of these questions are left unanswered, you may place it not applicable or you cannot find any literature. Would be also plausible. But kindly make, or at least try to uh, choose a pathogen with much literature present. Aside from that, the crop it infects, or the crops it infects, and disease testing and screening method for your pathogen place here. It's also on literature and documented sources of resistance. So is it wild races, germplasm, land races? Are there any literature saying that there is a source of resistance? And are the R genes identified? So is there hypersensitive response presence, and molecular markers for resistance present, yes or no, and of course, possible breeding and selection method that could be used. Okay? So this is just your toolbox. So based from here, all of these literature and information could help you decide on which approach you would try to use in breeding for your resistance of your insect pest and of your pests more likely. So for a moment, hello. Okay, sorry for that, an urgent call. So going back, you also need to answer the guide questions. So at what stage of development should test or assay screening for disease resistance should be performed? So I want you to also take a look in the literature. What stage, basically? So there's no definite answer, to be honest, in this type of question. So yeah, you may be surprised of what you will find out when you try to search on related to this question. And how does the following affect your screening or breeding or selection for disease resistance? If disease cycles are monocyclic versus polycyclic, and of course pathogen and lifestyle. If it's biotrophic, necrotrophic, or hemibiotropic, or it's facultative biotrophic, so on. It depends. Okay, so that is just and of course the narrative report. Now that is all for our module discussion before we start with uh, our presentation this afternoon. Does anyone have any question, or do you have any questions? So again, for the video recording on rice breeding, it has already been uploaded last week. So this week we also try to record the video this afternoon and try to upload it as soon as we can, or probably next week. Okay. So aside from that, the submission of the exercise for this week on exercise on worksheet 7.1 would be I placed no due date, but at least a week before the semester, and so that I can still check. Okay, so we will comply with the leniency, the memorandum calling for leniency towards the students in our exercise this afternoon and the previous exercises we've already done as well. So you may submit it as soon as as you have completed the exercise. Okay, just make sure to submit at least a week before the class ends so that I can still check your your uh, outputs. Okay, so I don't want you as well to be anxious or uh, to be afraid of going into class just because of all the that you have not delivered the deliverables present. Okay, so I just want you to learn, sit back, and of course try to enjoy the lecture and ask questions if there are. Okay, so are there any questions from the class? Yes.
So I have one. When is the last date of?
Sorry.
Uh,
Sorry, I was actually the volume was turned on off. Sorry. Please reiterate.
I was asking when is the last date of the class holding, which you are telling that we
Okay,
have
the
to.
last date of holding of active classes is June 11. So a week before June 11 is preferred. Okay, although I can still hold until June 11, but of course, uh, I do not recommend because it might be much more difficult for me to check your exercises. Okay, so try to
Okay.
submit it a week before June 11. Okay,
Okay. Thank you, sir.
you're welcome.
And sir,
Mhm.
what
Mhm.
about the presentations on NCTs, which we have already submitted? But are we
Yes.
going to have
For
the
the for the previous exercises, since the memorandum is only, I think, was only just published around exercise 5, so I'm not so sure if it is applicable for the previous exercises. So for exercise 5, 6, 7, 7, I can vouch that you can submit it anytime, uh, uh, during when as soon as it's available, for exercise 6 and 7, particularly for those exercises. Now for exercise 1 and 2, I know that, yeah, it might have been difficult. There might be difficulties for exercise 2. uh, I am willing to still accept submissions. Okay, so I will be checking them. So do not worry, do not fret. I mean, I don't want you to be anxious and uh, be filled with emotional concerns that will plague your mind. Try to answer it whenever you can, of course, but submit before the deadline. Okay,
okay? Thank you, sir.
you're welcome. Any more questions? Okay, so yes, okay. So I missed you guys. So I haven't seen you for quite a long time. So today I will be uh, introducing our guest lecturer for this afternoon. Okay, so I hope our guest lecturer is already. Uh, Wait for a moment. Uh, I think he already had the expired link. Copy joining in. Sorry from Okay, so let me introduce our. We have our guest lecturer for this afternoon, actually. So I think he is already here, and we are very lucky to have him this afternoon. So let me just read his curriculum vitae. Now, Dr. Mark Balendres leads a nationally recognized research group and is the laboratory head of the Plant Pathology Laboratory of the Institute of Plant Breeding, College of Agriculture and Food Science, University of the Philippines, Los Baños. He works across a wide range of economically important crops and seeds and a diverse plant pathogen, including bacteria, such as bacterial wilt of Solanaceae and bacterial stalk rot of corn, protozoa, such as the potato powdery scab, fungi, which is such as vegetable and fruit anthracnose, and mycotoxigenic fungi, and of course, viruses, including viruses of banana and vegetables. Research outcomes include the elucidation of the ecology and epidemiology of plant diseases, selection, and of course, very important for our exercise this afternoon, the development of disease-resistant crops, the development of diagnostic and disease-resistant screening protocols, which is one of the objectives of your exercise rather, and development of sustainable disease management strategies. Mark, Dr. Mark is responsible for many new records and some new pathogen reports in our country. He also identified several vegetables and fruit genotypes with resistance to various diseases. Dr. Mark, or Mark, simply Mark, is well published with more than 30 peer-reviewed papers in only the last 5 years, with a Google Scholar H index of 7 and with a total citation of 169. He has successfully won over 40 million competitive research grants and has won several awards for his research activities, including the Philippine Phytopathological Society Incorporated Geo Ocphemia Outstanding Plant Pathologist Award in Research. Wow, as its youngest award recipient ever in 2019. Recently, he received the 2021 UPLB Outstanding Researcher Award in the senior category for natural sciences. Okay, so I won't, without further ado, let me welcome and let's give a warm welcome or a round of applause, an online round of applause to Dr. Mark Angelo O. Balendres. Thank you very much, sir, for gracing us.
Yep. Thank you very much, Sir Rio, for that very generous and kind introduction. Um, so I've been informed by Sir Rio that there will be English-speaking students in this group. So I will have, for the benefit of those who will not be able to understand Tagalog, I will be speaking in English for this presentation. Um, I will now share my screen, Sir Rio.
Thank you very much, sir, and please go ahead.
Thank you. Yes. I have to. Okay, I have to find that. Okay. So, could you please let me know po kung tama po yung nakikita ninyo? Kung presentation slide na po. Hindi ko po kasi kayo makikita kasi I have to look at the presentation. So,
Yes, sir.
okay na po?
Yes. Yes.
Okay. All right. Okay, so this is not a very long it's not too many slides, but probably the discussion would take us longer, but relatively won't probably take us more than 1 hour for this discussion. I have to wear my mask because I'm sharing the office with my colleagues here. But anyway, so the vegetable pathology work at the Institute of Plant Breeding is a collective effort of various researchers with expertise ranging from plant breeding to plant physiology, and of course, in our lab would be plant pathology. In this afternoon's presentation, I will be discussing and presenting to you a very brief introduction to the general methods and practices in biotic stress resistance screening here at the Institute of Plant Breeding. And I'd like to thank Sir Rio for the opportunity to share with you all the research that we do here at the Institute of Plant Breeding. I would definitely welcome questions and clarifications, and maybe if you do have information that could add up with the information that I've presented, then feel free to add that later on. So in this presentation, I divided this to 6 major groups. The first one is to briefly look at why do we have to study plant diseases. Second is the general management strategies for plant diseases. And then we'll touch on where do we find disease-resistant plants and how do we know that they are resistant or susceptible. And then in the fourth and the fifth would be focused on the different screening procedures, one for bacterial wilt and one for fruit anthracnose. Later on, I will discuss why I've picked bacterial wilt and fruit anthracnose as model diseases for this afternoon's discussion. And lastly, we have to, more importantly, we have to touch on the factors that affects the disease resistance screening and what should we consider when we're looking at resistance and susceptibility of these plants. Now, what are plant diseases? Basically, diseases are any dysfunction or malformation in a typical organ of a plant. It could be caused by abiotic or abiotic stresses, more commonly by biotic stresses, and they are more contagious. The abiotic stresses are not contagious. So the biotic stresses are those caused by fungi, bacteria, nematodes, and viruses. And they are important to us because they reduce yield, the income of the growers are affected negatively, and also it impacts the quality of our produce. For instance, for fruit anthracnose, it has an impact on the marketability of your fruit, and also it does have an effect on the food safety as well, because once the fruit is infected with a disease, it opens up to a whole bunch of opportunistic pathogens like bacteria and fungi that could, for instance, for fungi that could produce mycotoxins, and mycotoxins are carcinogenic chemicals or metabolites that, when taken in a significant amount, could cause cancer. So the last one is that it could increase the cost of production in your field. Sometimes you could control diseases by adding a lot of nitrogen, not for all diseases, but you could also use um, biological control agents and chemicals as synthetic chemicals to control these diseases. And they're not cheap. You have to buy them, and that might cost you a significant amount of money. When you deduct that from the income, then it is still considered a yield loss, because that equates to the money that should have been a yield rather than a cost for fungicides and fertilizers. There are different ways or methods that we could do to make sure that diseases don't go to the field, or pathogens don't go to the field, and that diseases don't um, or the disease don't it will not be considered as an epidemic. So different methods according to Wetzel. There are four general principles. The first one is exclusion. So these are measures that will prevent the introduction of your pathogen into your field. So you make sure that there are phytosanitary measures to make sure that the disease plant um, does not reach the field where there is a healthy plant. The second one is eradication. These practices are being done when the pathogen has already been established in your field. So, for instance, if exclusion has failed in that particular farm, then you will have to do some eradicative measures. The third is protection. These are considered as barriers or practices that would avoid the contact of your pathogen and your host. For instance, mulching is a protective measure because it provides a barrier between your pathogen in the soil and those of your fruits that are hanging in your plants. So the pathogen is established already, but you want to avoid the contact of the pathogen and the host plant. The last, and I think the most important among the four principles, is resistance, because this is the most durable one. This is also the most sustainable and relatively cheaper, because you don't need to hire people to do mulching. You don't need to hire people to do other cultural practices. So with resistance, you just need to buy the seeds of the plants that are already considered that are able to resist the presence or the infection of the pathogen. Now, in plant pathology, there is really no single effective control measures. Even though you have a resistant plant, it is always a good practice to combine it with one or more of the other principles that I've mentioned earlier. So if you do make sure that there are barriers or there are phytosanitary measures that you did in your farm, and at the same time you keep on monitoring or surveying the farm, doing some soil solarization, also removing infected plants, and at the same time planting resistant or less susceptible variety, then you are likely to mitigate the impact of the disease. And if there will be an epidemic, that would be these integrated practices would be able to lessen the damage that this disease would have in your farm. So you would be able to delay the onset of infection, and by the time that there is an epidemic, you've already harvested some of your produce. So integrated is still the best way to combat diseases on your plants. Now, I'm highlighting resistance here because for the purpose of this class, we'll be looking at how do we deal with diseases in terms of resistance, and we would be looking at different methods of selecting for these resistant plants. But the question is, where do we find, or how do we find these resistant plants? So there are five major, I wouldn't say categories, but five major methods or sources of your resistance. We can always go back to the wild relatives of your cultivated plants, and this is why germplasm collection is a very critical component of disease resistant screening, or developing for disease resistant plants. And I think Sir Rio could justify that how really important collecting wild relatives of plants in the country is critical to our plant breeding work. Second, we could do standard breeding procedures. So you could use the wild relatives as your parent materials, or you could find something else by crossing your wild relative and a commercially available variety. So you do selection and hybridization, so crossing one plant with another to come up with your F1s. The third, or the first method, this other method probably is through induction of mutation. So you use chemicals that we call mutagens, and these chemicals would be able to tweak a little bit of the genes and the DNA sequence. That particular tweaking there would result to a change in the phenotype, or probably a change of a particular trait. And that's very exciting because sometimes it's a trial and error. And this means that you could find something that is really good at the same time. You'd probably find the worst traits that you could find in a particular crop. And this is more of an explorative stage as well. Like standard breeding, you'd probably have a ratio of 3:1, or 9:3:3:1 expected ratio. But with mutation, it could be something more different from that of the standard breeding. But it's very dangerous as well for a researcher to do mutations and you're going to use mutagens, because it's dangerous for people to be exposed to these chemicals as well. So you have to handle with care if you're working with mutagens. You could also work on different activators. So some of these chemicals can be immediately exposed or used to a particular plant, let's say a susceptible plant. So there have been researches about using different plant activators like beta butyric acid, salicylic acid. So you just need to spray them to these plants, and these plants are being induced, and this response is called systemic acquired resistance. So the plants are able to develop this kind of resistance instead of being from susceptible to being a resistant plant. And the last one, and I think many of you as plant breeders or agronomists know, is genetic engineering. So you're trying to manipulate the genetic makeup of the particular plant by either inserting, deleting, or adding something or removing something from this particular plant or plant genome. And that would be that would undergo different biosecurity measures or protocols to identify which one is genetically engineered and which one is not. Right. But regardless of the methods or your sources of these resistant plants, all of these methods would need to undergo what we call phenotyping. This is basically just a method where you need to assess which plant line or genotype is resistant, moderately resistant, or susceptible. And I think this is the main chunk of our presentation, which is on identifying those resistant and susceptible lines, and what are the factors that we need to consider to identify these lines. So you can't just plant these materials in the field and then there's no disease, and then you would say this plant is resistant to a particular disease. And so it could just be because there is no inoculum in the field. That's why there is no infection. So there are different factors that you need to consider when you're looking for resistant plants. Now, in this presentation, we'll be looking at two different pathogens. bacterial wilt, which is caused by Ralstonia solanacearum, and anthracnose caused by Coledotricum species. We use these two um, diseases because they have a very complex organic microorganisms that cause them. So one is a bacterium, one is a fungus, and they're complex because it's not just one species that causes the disease, but there are different species. So we put SPP for different several species, but for one species you would just put in SP. Bacterial wilt attacks the plant. The anthracnose mainly attacks the fruit, but there are species of Coledotricum that attacks the oldest leaf of the leaves of the plant. But we'll focus on fruits here. So the bacterial wilt is a root pathogen and anthracnose is a foliar pathogen. The bacterial wilt enters the root. The anthracnose enters to the surface of your host plant, for instance, the leaf or the fruit. The other one is the pre-harvest disease. The other one is a pre and post-harvest disease, which is the anthracnose. And so bacterial wilt is systemic, which means that once the plant is infected, you're likely to get infections as well from the other parts of the plant, or you could get the bacteria from the other parts of the plant. With anthracnose, it's more contact. You could only get the infection from where the spores land, not on the places where um, there are no visible infection or visible um, signs of the pathogen. So this is why it's good to to use these two diseases for comparisons for your screening, because you would see that not all diseases should be treated equally and that different methods are being used for different diseases. So I'll focus on bacterial wilt and anthracnose. So we'll start with bacterial wilt, and this will be the overview of the bacterial wilt screening. This is the methods that we use here at the Institute of Plant Breeding. And some of these methods are also being used by different organizations and institutions around the world, looking at the resistance of tomato, pepper, and eggplant to bacterial wilt. So we start with your pathogen, your test plants, your inoculum. Then you could go to the lab for in vitro assays, do some glasshouse assays, and later on we'll discuss about how do we assess whether a plant is resistant or whether it is susceptible. For the bacterial wilt screening, the first stage is to have a knowledge or information about your pathogen. And for non-plant pathologists, it could be very difficult, but there are several ways that you could do to ascertain which one is bacterial wilt or which one is not. So if you have a wilted plant, usually bacterial wilt, the youngest leaves, the upper portion, is mostly the ones that are being infected and the ones that will start to wilt. And with bacterial wilt, your plant doesn't turn yellow. It remains green, maybe turn yellow later on for severe cases, but usually it should be green all throughout. If not, then that could be caused by Fusarium wilt. So you cut the base of the plant and you'd see some discoloration in the vascular area here. And when you put that particular plant or tissue in distilled water or any water, clear water, you would see a whitish substance coming out from this infected plant. And that is actually your bacterial ooze. And we call this one the bacterial ooze test. And this is the most reliable, probably for bacterial wilt, easiest, most reliable test for bacterial wilt in the field. And once you see those viscous milky white substance, that indicates that there's a bacteria on those infected tissue. And for us, once we found this whitish substance, we could get a sample from this particular water and then streak that on a culture media, and then we do molecular characterization. For bacterial wilt, we need to do molecular characterization because it's very difficult to identify which one is Ralstonia pseudosolanacearum and which one is Ralstonia solanacearum. So these are two bacterial wilt species, and they can infect tomato, eggplant, and pepper in the Philippines. Right. So you already have your inoculum, you already have your bacteria, and you know that there is a disease in the field, and you wanted to check for materials that has resistance on this particular disease. So you start sowing your seeds. You need to sow for tomato. You need to sow them because you sow them 7 days before your test, or I'm sorry, 3 weeks before your test. So transplant them 7 to 10 days. If you're doing an in vitro test, which I will discuss later on, or step 4.1, you need a 7 day old seedlings for your test. But if you're doing a glasshouse spot assay, you need 3 week old material. So you need 2 more weeks after you transplant. But for this in vitro assay, you just need a 7 day old seedlings. This is step 3 here, which is the preparation of the inoculum. So you have 48 hours of culture in Kelman's media. So it will look like pinkish appearance in the media, and then you'll have to select a single colony. So this is already purified, but the test will require you to get a bacterium from a single colony. So you just pick one that looks really Neurostonia, and then you strict that to a new Kelman's media, but without tetrazoleum. So this material will now um, it will not show any pinkish appearance already. So within 24 hours, it will be just whitish appearance. It's just like this one. And then you standardize that using a spectrophotometer, and you will need one. Right. So Rio, is my internet okay? Okay lang po ba?
You were disconnected for a few seconds lang, sir.
Yep. I'm using my mobile data, and every time someone calls, it stops the internet. So
Yes.
that's good.
Yes.
Right.
Yes.
So let's continue. So once you prepared your test plans and you've already prepared your inoculum, we will then proceed with the different screening procedure. And they'll start with in the laboratory. And this is an in vitro assay. Now, in vitro assay, before I forget, you need to make sure that you also planted your controls. So for bacterial wilt, we use the tomato cultivar yellow plum, and resistant line, we use the line H7996, which is a Hawaii line that has resistance to the bacterial wilt. But this plant doesn't have a commercial merit because the fruit doesn't look like something that you would buy in the field, or sorry, in the market. So we use that as a resistant check. So they need to be planted at the same time with your test plant. So if you have 20 lines, then you will have a total of 22 lines. And also you could also add a cultivar check if you're using wild genotypes or F1s or anything. um, it would be nice to add a cultivar a check, which is a variety of a particular crop that is being sold in the market. So that will be your varietal check. Now, in this in vitro assay, if you have a healthy tomato seedling, you just need to submerge that particular root of the plant into your inoculum suspension, which is 1 million spores or a CFU per mil. And then you put that after dipping in this suspension, you will leave that for 5 minutes just to air dry. And then you add a microfiche tube, a water in the microfiche tube, and then you incubate that in a growth chamber. Usually you'll get wilting after 3 days, depending on the reaction of the plant. Now you will ask, why do I have to leave this plant for 5 minutes in exposed to air? Why not just immediately put that in the water? Well, there is a study that says that finds this particular method as more efficient in screening for resistance, because the mechanism is still unclear, but these aerodynamics there, the ability of the bacteria to be imbibed by your plant when there is no water increases the chance of getting an infection. So when the plant is resistant, don't get infection, but if it's susceptible, then you're likely to get infection after 3 days. So you need to follow the procedure. There's a reason why you don't have to put the plant immediately into the water. You have to expose it first for 5 minutes in the air and then add the water. The second method, which is the most common, is the glasshouse spot trial. Here, you prepare a 3-week-old or 3 to 5 leaf stages of your plant, depending on your lines or varieties. Before you inoculate, you make sure that you don't water the plant or the plants 24 to 48 hours prior to inoculation. For small pots, you could add 30 ml of suspension. For large pots, you could add 50 ml of your inoculum suspension. And then after 24 hours or 48 hours, depending on the need, you could water the plants you can start watering the plants and water it as needed. And then you need to leave them for 4 weeks after inoculation. But the data gathering would be 7, 14, 21, and 28 days. I'll discuss more about the data gathering in this particular slide. So these are the data that you needed when assessing for bacterial wilt resistance. Of course, you need to take photos. That's very important because you're trying to compare the wilting of the plants. But these are the basic data needed for bacterial wilt screening. First is disease incidence. So this is just the total number of plants infected over the total number of plants assessed. Usually we do minimum, this is minimum, 5 plants per replication, and each line will be replicated 3 times. So you will have a total of 15 technical replicates. That's minimum. So you could go to 10, 15, or 20 depending on your materials. The second one is disease severity. So it is based on a rating scale, which I'll show you later on. And this is just a proportion of the disease tissue or plant, and in terms of the vigor and the whole plant stand of your materials. The next is the days to first infection. So you need to note when was the first or when was the day where you first observed the wilting symptom. And then you also have to get the number or the days to 50% incidence. So when 50% of your plants have already wilted, and then you stop there for this particular data, and then you use disease index. So this is where we base the susceptibility or resistance of your plant, and this is just computed based on the rating scale. Later on, I will show you that. And more importantly, the time. So you need to know which day or week you are going to get the data. Usually for us, we do 7, 14, 21, and 28. So that's 4 weeks, because sometimes there are plants that would wilt after 3 or 4 weeks, and others are just really very susceptible. They could wilt at 7 and 14. But of course, aside from the 7, 14, and 21, you need to get the data for days to first infection and the days to 50% incidence. Now, you need to remember that before you do the assessment, you need to water the plants at least an hour before assessment, for the reason that it could be just because there's a lack of water on your plants. That's why it's wilted. So you make sure that you provide water first, at least 1 hour before the assessment. Now, there are plants that would be asymptomatic, or there could be plants that were not wilted, but if you check the stem, the one just above the root, or above the soil, you could still get infection, or you could still get the bacteria. So we do some randomly we check those materials that are resistant based on the scale, and we still get bacteria in those stems. That simply means that it could be that the material is more of a tolerant than a resistant, which means they tolerate the presence of the pathogen, but the pathogen don't cause significant wilting on the plant. So it's always good to check this particular um, test to make sure that you're using the proper term when you're reporting it with someone else. And if there is a reviewer who is really good in bacterial screening, they might ask if there is a bacterial colonization in your stem. So you can do bacterial ooze testing or streaking the bacteria from the ooze or the dilution to your culture media. So this is the disease assessment scale or the severity scale, and we used the Winstead and Kelman, which was published in 1952. Now, before using this scale, we compared 3 different scales used for bacterial wilt, and we found out that this one is the most conservative one, which gives us a very few resistance, while the other 2 rating scales, they were giving us a lot of resistant plants, but they're not really resistant when you test them in the glass house. So um, we're working on a paper right now about this comparison of different rating scales, and so we're using the Winstead and Kelman. It's a very old scale, but we found out that this is probably the most or the best scale to use for resistance screening. This is also the scale that's being used by the World Vegetable Center in Taiwan. So you'll do a little bit of math, but it's not very complicated. You just need to put the numbers. There's a formula, so you just need to put the number of plants with a rating of 1. You add that with the number of plants with a rating of 2, and then divide that with the total number of plants observed, multiplied by the highest rating, which is 5. And then based on that disease index, that's per line. So you'll get their disease index of 0.0 to 0.2 until 1. So it can be converted into percentage if you multiply by 100. So those with 0.0 to 0.3 are considered as resistant, and then 0.31 to 0.40 would be moderately resistant. For us, we always go with the highest resistance level as our parent material, but we still consider those that are resistant and moderately resistant because they might have some traits that are very important or commercially preferable. So we do keep them. We don't just focus on the highly resistant one. But if you find some very susceptible plants, but notice that the fruits are really high yielding, or the plant produces tons of fruits, then also keep them, because you could always cross them with a highly resistant plant, or you could use the highly resistant plant as your rootstock and use your susceptible plant as your scion. Right, so this is an example of the trial, the first glasshouse trial for the World Vegetable lines that we have in the lab at the moment. So we have a project funded by the Department of Science and Technology in collaboration with the World Vegetable Center. And so for the first Ralstonia species, which is Sojus solanacearum, we did identify several lines that are considered as resistant. So your resistant check is here and your susceptible check is here. So when you do presentation, or when you present this work to your colleagues or to your supervisor, you make sure that you plot in the resistant and the susceptible check to the extremes, and you'll find the materials that are with close to resistance and the close to susceptible check. So it's always good that you put the references in there. It would be easier for you to discuss, and it will be easier for anyone to see which lines are considered as closer to the resistant and closer to the susceptible check. With Ralstonia solanacearum resistance, we also found several lines, and what we wanted to do here is to pick the ones that are resistant in both bacteria. And you see that TM011 here, and TM015 are our candidate lines. A bit of a incidence here for TM011 and 015, but not really very significant compared to those lines that are very with high incidence or high in disease index. So most of them are generally resistant, but only a few were highly resistant. So we'll focus on lines that we could use as our parent material crossed with any materials that are considered highly high yielding or high lycopene content. So all of these traits that are preferred in the supermarket or preferred by nutritionists. Okay, so we're done with bacterial wilt. Probably we'll do the question and answer later on to just continue with this one. So here is for fruit anthracnose. So we're dealing with fruits now. Earlier we dealt with a plant, but this one is on fruits. It's the same overview. It's the same strategy. You have to find your pathogen source. You need to prepare your test fruits or plants. You need to prepare your inoculum. You do detach assay. You do glasshouse assay, and also you do assessment using a variety of rating scales, and that depends on your disease as well. For your pathogen source, we usually collect those materials in the field, and then we do a polyphasic approach to identifying the pathogen. With Colitotrichum species, it's very complicated than the bacteria. Here you will have more than 200 species of Colitotrichum. And so we need to be very strategic in how do we identify them. So here we combine morphocultural characterization with pathogenicity assay and molecular characterization. So this is a very long discussion, but summarize here in this diagram from disease observation to DNA sequencing. I'll probably just post this in Facebook so Sir Ryu could have an access and he could provide this to you. But please do ask any questions later on if it's very complicated or if you have any questions about this one. Now, after that, if you have already identified the pathogen, you make sure you have a backup and you purify this for preparation for your next trial on disease screening. Now, how do we prepare fruits? You could buy the fruits in the supermarket or you could grow your own. For pepper, we usually grow our own pepper because the ones in the supermarket, they are highly immersed or exposed to fungicides, and sometimes we don't get infection because of these fungicides. We could clean them anyway if we don't have any plants in the field, so we usually just buy them, these fruits, and then just really wash them properly to remove all the excess fungicides. So here's an example of a tomato trial for anthracnose. Here's a Averola trial for Neocytilidium, and then we have multiple fruits here for our Colitotrichum pathogenicity test as well. You could leave a mark, a circle into the area where you will inoculate, but you don't usually put circle for anthracnose because it will be overlap later on with the lesion anyway, but depending in how you will, but you have to make sure that you know where you inoculated it. So a point will be fine, or probably a little bit, just a little circle, but not too big. And how do we decontaminate the fruit? We just immerse in tap water, and then we have 10% Zonrock solution, and then wash 3 times in distilled water, and then air dry or blot dry in sterile tissue paper. And so now we will prepare your inoculum. So for fungi, you need to prepare 7-day-old culture. You harvest the spores if you have spores. If you don't have any spores, then you use the mycelial plug. So a 5-millimeter plug that will be used for inoculation. And then you standardize that for fungi. Higher concentration is needed. For bacteria, I mentioned 1 million, but for fungi, you could still use 1 million, but 10 million would be the most appropriate one, which is 10 to the 8. spores per mil. For bacteria, it's 10 to the 6 CFU per mil. But for Colidotricum, they're very destructive, so 1 million would be sufficient. But for publication perspective, you should use 10 to the 8, which is 10 million spores per mil. So this is one of our research associates, Darby, and he's inoculating dragon fruit. So you see here that there is a wounded and the unwounded part, which is in the top portion. So he is inoculating suspension to this wounded area, and then incubate in the Tupperware, which is airtight. That means there's a humidity that would enhance the infection process of your pathogen. So the biology of your pathogen, knowledge of the biology of your pathogen is important when you're doing selection trials, because you need to know what conditions favor the development of the disease. So this is an agar plug or mycelial plug method. So there are pathogens that do not produce abundant spores. Sometimes it's disappointing that after 14 days you don't get spores. So what you could do is just use the mycelial plug as your inoculum. You just bore a hole for 5 mm and then place this mycelial plug on your predetermined area, whether it's wounded or unwounded. So this is not on fruits, but on the fluorescence of a dragon fruit, and we've inoculated the ones in there. The green part and the white part as well. And you could do the reason why we do detached fruit assay for anthracnose is because anthracnose is a pre and post harvest pathogen. You could get infection in the field, but then they develop in the glass in the supermarket or in your fridge. So you could always test them not in the plant and detached from the plant. But for resistance screening, it's still advisable that you do resistance screening under a more realistic condition, not necessarily in the field, because it's not good to spray your pathogen in the field for the simple reason that you might be producing more spores or providing inoculum for other crops in the succeeding cropping season. So do a glasshouse trial on the plants. You could do spraying as well. So no need to wound, but if you do wounding trial, you could just prick the fruit with a needle or a yellow tip or a white tip, and then spray it with your inoculum, which is standardized as well. Just make sure that you evenly spray it, not too much in the other line and too little in the other line, so that would be a bias. Similar to bacterial wilt, we use a screening rating scale, and here we use a rating scale by Morrell et al. That was used on what do you call this one? Forget the olive fruits, and you can use it with any fruits anyway because it has been standardized. So 0 to 1 is considered resistant, and then moderately resistant for 2. And this is based on the fruit surface infection. So the larger the lesion, the more susceptible the plant or the fruit. So you have number 5 as you're very susceptible. So this is an example on banana, and this is Colidotricum musae. So we use 2 fungi here, and only fungi or fungus 1 had this bigger lesion compared with fungus 2. So simply means that this fungus 1 is more aggressive and virulent on banana, but the fungus 2 is not. So there is this specificity of different inoculum that someone would use. So you need to know which one is more aggressive as well when you use a resistant, or when you conduct a resistance screening, because if your pathogen is not aggressive, then you might not get any infection, and that causes a problem with your screening. That's why controls are very important, and I will discuss that later on. which I think is the next one. Sorry about that. Um, so these are the critical factors that you need to consider when you're doing resistance screening. Your test plant or fruit, your pathogen, your environment, the time, the data, and your controls. And we're going to touch base on these particular factors. The first one is test plant. You need to consider if it's already considered resistant or susceptible, and you need to use, or you need to. You don't have to know if it's resistant or susceptible, but you need to know which stage of the plant is more likely to get infection. So here, the age of the plant host is critical. So an example for banana anthracnose or banana rotting is this one. So infection occurs on ripe fruit. So if you do inoculation on ripe fruit, then you're likely to consider all your materials as resistant. So that is not good because you'll always get resistance because it's unripe. So you need to know whether it's ripe or unripe. If you don't know, then go to the literature and read information about previous work. If it hasn't been done, then you could always do your own trial. So it's true with leaves as well. You need to know whether it's young leaves or older leaves. Some trials would use second fully expanded leaf, and others would use the most mature leaf. And also, you need to know whether you're going to use a 1 month old seedling, a 3 month old seedling. So you need to consider this information, and you can only know this if you read papers related to this particular biology of your pathogen or your disease. Next, you need to know whether you get infection when it's wounded or unwounded. Now, there's always this argument that you will always get infection when it is wounded. And that is true, but not all true to all pathogens and disease. The aim of the wounding is actually expediting the process, because in the field, even though you don't wound them, there is always this, there are insects, there are physical factors that would lead to wounding. So you are trying to include factors here that would induce or that would allow the pathogens to enter your host tissue. A good example is fruit anthracnose caused by Coleotrichum. So here, only the fungus number 5 caused infection in both wounding and unwounding. So other species here, species 1 to 4, they can only cause anthracnose when there is an entry of the pathogen. And this is still an important information because when you're handling your fruits, you might be able to cause wounding in these fruits, and that would allow the pathogen to enter. On pepper, it's a very different story. Species 1, species 3, and species 4, they were able to cause anthracnose in both wounding and unwounding, but there is a degree of variation. So for me, I would use species number 3 as my test materials for selection for resistance. And the Carabao mango has been always used as a susceptible check, and the cultivar Django for pepper has been used for anthracnose resistance as screening here. But you could always find there's a lot of susceptible plants anyway, so you could use them. What is important is what variety or cultivar you're using as your test host or host plants. Next is the variety or the genotype. So that's what I mentioned earlier. You need to add susceptible and resistant check when you're doing resistance screening. If you don't have resistance check, just make sure that you have a susceptible check for comparison. And then you also need to know the size and the shape of the plant material that you're going to use. And the single reason is that you need to get the lesion size, and sometimes it's the surface area. If you use ImageJ, that will allow you to get the proportion of the lesion that has the disease and those that are still healthy. Now let's go to the pathogen. You need to know that the pathogen is very virulent or aggressive. That's why if you have multiple species or isolates, you need to test them first, which one is most aggressive or most virulent. However, if your test is to know the adaptability of your material to a different geographic area, then you might as well test different pathogens from different area. So you need to check the aim of your experiment. Are you testing it with different isolates from different area, or are you just testing it with the one that is most dominant, most virulent, and most aggressive? So also you need to know whether you can purify your pathogen. You cannot test or you purify something if the pathogen doesn't really grow on a culture media. So you need to check for this, and also whether they produce spores or they don't produce spores. This information needs to be considered. Inoculum density, I mentioned earlier that for fungi, it would be 10 million spores per meal. For bacteria, at least 1 million CFU or colony forming units per meal. You we usually you need to standardize that so that next time you'll use the um, same concentration. But if your purpose is just really to know which one is pathogenic or which plant could be susceptible, you don't need to standardize it as long as you see somehow a lot of spores in your um, sample. So if you do a microscopic test and you counted 100 spores in that particular field, then that's perfectly fine. That's already more than what you needed. So no need to reduce that to 10 to the 6 or 10 to the 8. If the purpose of the experiment is only to look at pathogenicity, not comparison of different isolates and only comparison on different types. Right, so repeat. Here's another example on the effect of the source of your inoculum. So these are different Gliosporoides species or isolates of Gliosporoides, and they were obtained from different crops. And the results here indicate that the mango isolates is more virulent and susceptible to mango. But the pepper, then the rubber tree isolate, they're not as aggressive. Aggressive means um, the days to first infection. Virulence means the degree of infection. So after a particular date, let's say 12 days, which one is more virulent, the ones with the biggest lesion? If you're talking about aggressive, which one infected first? So here, mango and the days, the 6th day, it's the mango and the guava isolate, probably a little bit of grubber tree, but you notice that pepper very minimal, and then later on after 9 and 12 days you notice a progression of the disease, but the mango isolate is more virulent than any other isolate. The guava could be as well, if you compare that, so there might be no significant difference between mango and guava. But what this um, knowledge is telling you is that you need to use an isolate that which came from your test plan. So if you're working with tomato, it is good to use tomato isolates. But if you already there's a asterisk there, unless of course you have already proven that different isolates had no effect on host. So if you have tested that the guava isolate had no significant difference with the mango isolate, then you could use guava isolate if you have more spores in your guava isolate. So it's a matter of proving that there's no effect or there is an effect. So make sure you have these types of data, or otherwise you need to do it on your own. Right, so age of inoculum is also important. So this is a paper we published in 2019. And what we found here is that if you have a 1-year-old culture in the media and you use that compared with a 7-day-old culture, you don't get much of a disease on your fruit. So you see here that the lesion length is at probably 0.5, and then you'll get 4 or 5 for a 7-day-old. So this simply means that there is an effect on the age of your inoculum, and you need to re-isolate the pathogen first. So the solution to this is when you have a 1-year-old culture, you inoculate that in your host plant or fruit, and then re-isolate that. So once you re-isolated that, use that as your um, inoculum for your trials. So the second graph here shows you that when you re-isolate the 1-year-old culture here, they become more virulent than the 7-day-old culture that was not introduced to your host plant. So there is this interaction between the host and the pathogen, which activates the pathogen's um, virulent genes. That's why they become more virulent when they encounter the host at the second time. Um, so that's the whole message of this paper. You need to reintroduce first your isolate that was stored for a long time, probably 3, 6, or 1 year, 6 months or 1 year, and then use that as your inoculum, the re-isolated one. For the environment, so you need to consider the temperature, humidity, and soil temperature. If your pathogen is a temperate pathogen, which means below 15 degrees, and you grow the plant in 30 degrees, then it's likely that you don't get infection. So you need to know the temperature that your pathogen is ideal for growth of your pathogen, and also you need to know whether the pathogen needs higher or lower humidity. That's why we incubate our fruits in the your chamber in a airtight container, because spores love the humid condition, and they do sporulate when there is a lot of water in the in the atmosphere, in the air. And also for bacterial wilt, as it's soil moisture is important. That's why um, for inoculation, you need to make sure that it's not watered, because if there's too much water in your soil, then the roots might imbibe or might um, absorb the water and not the bacteria. So with the addition of bacteria, when the soil is dry, then the plant will absorb the bacterial suspension. So after that, you need to moisten the soil again, so that there will be a flow of bacteria in the soil and towards the roots as well. The environment, so this is just an example of the ones that I mentioned earlier. We do place them in the same container. So these are two different trials. So you need to make sure that you have the same size, because of course the smaller the container, you'll likely get more humidity as well, and but more sensitive to temperature, outside temperature. But for the bigger one, you see that there's a difference with a smaller one in terms of the production of abundant moisture at the edge of the container. So it's always good to use the same size and the same brand of containers, particularly if you're publishing, because it might have an effect on the outcomes of your experiment. Now, for your time, you need to know whether are you doing daily, weekly, or monthly assessment. Also, for your data, I mentioned earlier, qualitative data is just plus minus, which is positive, negative, or disease or not. But for screening for resistance, you need a quantitative data. That's why you're using disease rating scales. You're measuring the lesion of the disease in the fruit or the leaves. And those scales are being used to convert into a disease index, which will give you indication whether it's resistant or not. And this is only going to be successful if you have a control, a proper control. So you need to have a positive and a negative check, which is resistant and susceptible. Internal checks, we usually use plants without water, and or plants inoculated with water, sorry, or a mock experiment where you could just inoculate it with whatever it is that is not bacteria or that is not water as well. So usually just the checks and the internal are the ones that are more important. You can always add mock just to measure the effect of water, which is your internal, on the plant productivity. So aside from resistance, you could already measure agronomic changes in the presence or absence of your bacteria. So it could be an additional data which could be used for your paper. And probably I'm going to end that with a statement here that your experiment is as good as your controls. And without the proper control, you're likely not to get meaningful data from your experiment. So if you don't have any controls, let's just say you don't know which one is susceptible or not, um, try to read papers on other crops that this pathogen can cause a disease. So you just really need to make sure you have something in there that would show a disease symptom. Because if all of them are resistant, all of them do not show disease symptom, that simply means that there's a problem with your pathogen. It's not your materials, it's not your plants, but it could be your pathogen. So you've wasted your time preparing those materials, but then realized that your pathogen wasn't really aggressive or virulent. And we do have a website at the Belanders Group, uplb at .wordpress.com. Please do visit us or follow or subscribe if you want to receive um, notifications about our research and also about plant pathology in the Philippines. These are the staff under the vegetables pathology group. So we have Herbert, Deborah, and Dr. Fe, and also Catherine from NPGRL. And also we have some support staff as well. And our research is funded by the Department of Science and Technology. And I guess that ends my presentation, Sir Rio. And I think um, it would be nice to hear um, what others or what our students think about the screening for resistance. But if you do have any questions specific to your thesis or your studies, please do ask. I'll do my best to answer those questions. If I can't, then I'll let Sir Rio answer the questions. Thank you.
So, okay, so thank you very much, Dr. Mark. For I would like to request everyone, if it's possible, can you turn on your screens? If not, if it is possible, so that Doc Mark can also get a glimpse of your beautiful and handsome faces. So aside from that, if ever, I would also, if there are any questions, kindly ask them right away. So, anyone?
Yes, sir. I have a question, sir. In your first slide, like you mentioned that the cost, like the least cost required for the plant is by attaining by making the crop resistant to certain diseases. Like, but suppose if we think practically, we cannot just buy the resistant plant and grow, and we can't do nothing. And I mean, like we cannot just leave it like that. So we need other. We also need to like apply other what to say practices for protecting against other diseases, not only the target diseases.
Yep.
So like it will like eventually leads to the like making the like doubling the cost. For example, if a resistant seed for like let's take an example for the BT cotton. Like if the BT cotton seed costs 100, then like uh, even though if the it is resistant, and
Mm,
we also need to add other practices, then it will cost more than the expected amount.
mm,
Then also even most of the I think most of the like transgenic crops have the terminator technology, which the private companies applied so
yeah.
that they need to buy another amount. So can you give some like idea about it?
Yep. Well, thanks for that. Your name is quite long, so I don't know how
It's
to.
Satya, sir. Satya is fine.
Satya. Okay. All right. So Satya Vanda. So Satya. I think you raised a very good um, long-term problem about this whole breeding process, where the cost of resistant seed is more expensive um, than your yield probably later on. But I think that's an option for the farmer to go into. And that's why I've mentioned that with integrated disease management, the farmer can opt to not use resistant varieties if there is no resistant varieties. If there is, they still need to consider whether can they be able to access them without affecting the potential yield that they will gain. So it's not simply that because there's a resistant seed, you need to use them. You need to consider you wanted to plant this material because you need to gain more profit. But if you're planting this and it's more expensive than just planting without resistant and using cultural method, then do the cultural method. So at the end of the day, it's going to be a decision that a farmer should make whether he or she is going to buy the seeds. And buying this from a, let's say, a private company would really be expensive. And if you're using a genetically modified one, you cannot keep the seed, and this is under the license. So you can't keep the seed and plant it again. So that's a good point. You raise a good point that why should I plant a very expensive seed and then later on I can't keep the seed, and at the same time I'm losing a lot of profit from it anyway. So there are other options, of course, and part of the research that IPB is doing is producing seeds that are open pollinated, that will allow the farmers to harvest those seeds that are resistant. The problem is that our enemy is more diverse, they're more destructive, and research has to go on because one plant can be resistant with a particular disease, and you're right, it could be susceptible to a different disease. So probably before you buy something, you need to assess as well what are the diseases you have in your field. Because if there is a field with multiple diseases and you have a plant that can only be resistant to one disease, then that is likely not a good decision to plant there. And probably the suggestion would be to find a different place. You have to leave the plant for several years, or you have to leave the area for several years without any crops so that there's a cropping break. So that would allow the interruption of the disease cycle. And with this interruption, the pathogens will lose its ability to produce more and more inoculum. But for bacterial wilt, that is very difficult because they can survive for 50 years without any plant in the soil because they have a resistant spores that will allow them to survive without their plants. That's why bacterial wilt is the most destructive soil-borne disease for major vegetable crops. And resistance is likely the only most durable method that we could get to solve or somehow mitigate the impact of this disease. So I hope I was clear with the explanation, but yes, let me know.
If I may add as well, Satya, it must be noted that resistance breeding needs considerable resources. So if ever there are alternative control measures available, such as pesticide treatment, disinfection of seeds, disease-free planting materials, when available and economical, should be considered before doing resistance breeding.
Yes.
So, yes.
Thanks for that, Sirio.
So, any more questions from the class? So, I think that's let's keep the ball rolling. So, I think this is a really good opportunity for everyone to ask about screening for disease resistance as well as resistance breeding. And we have our very great expert here in plant pathology and breeding for disease resistance, Dr. Mark. So, anyone from the class? Yeah. Yes.
Very quiet class.
Yes, there's a very quiet class.
Very quiet
Oh.
class.
I haven't seen them for 2 weeks and they're this quiet.
Okay.
Yes. Any more questions? Particularly, you can ask them about anything, even about things that on screening. Yes, since it is technically one of the major aspect in breeding for disease resistance is the correct way or the appropriate way to screen. Yeah. So anyone? So sir, how about sir, when you try to do field and glass house resistance, which takes precedence, or should the two be complementary with each other, sir, with regards to doing it in a doing a field resistance screening and a glasshouse resistance screening?
Mhm. Yeah, that's another good question, Sario. It really depends on how many plants are you going to plant in the field or in the glass house. So if you're just trying to plant those lines that are potentially a good candidate, so if like here we have, let's say 4,000 plants, then or 4,000 lines. So you can't just really plant them in the field or in the glass house. So you do in vitro test first. So you filter out susceptible lines, and then you do glass house trial, and then maybe from 100 you will have seven potentially high materials. So remember what you mentioned earlier, and that was really good. That you need to make sure that that what you're doing is economical as well. You can't just do trials planting everything and some already are resistant, are susceptible. So just make sure that you have one line that is considered susceptible, and the rest, probably those best lines that you have, you can plant that in the field. What you can do in the field is do uh, disease nurseries on those areas with high infection of this particular disease. So you plant those materials in those area. We don't spray the inoculum, or we don't um, inoculate additional inoculum in the soil, because that would add more inoculum, which is ethically, it might be a problem in the future, especially if it's a different strain. So um, if we have a strain from Mindanao, we don't test that here in Luzon in the field, because you might be spreading a different strain. So you need to consider that. So when you do conduct research, you need to make sure that you're doing it in such a way that you're not causing additional problem. So you're solving another problem, but yet you're here adding more problems. So back to your question, you can do glass house and field trial at the same time if you have resources. If you have multiple glass houses, then that's perfectly fine. If you have 2 hectares of land, that's perfectly fine. But if you have limited resources, then you have to strategize in such a way that you get a meaningful data from the available resources that you have. So if you're a student, probably make sure you could test only a handful of lines that are already pre-tested before. So you need to consider money, resources, time, and also um, other people's time as well.
In that line of questioning, sir, I'm just really curious. So, would this mean that there is appropriate timing as well as appropriate developmental stage needed uh, when you do field evaluation for disease resistance? So, particularly for different types of diseases with respect to their disease cycles and so on.
Mhm.
Yes.
Yeah, that's right. Because may tinatawag, we have something that we call um, early resistance or adult resistance. So you in bacterial wilt, usually those that are resistant when they're at the vegetative stage are likely to be resistant adult stage for bacteria. But there are diseases where the plants are susceptible in the younger stage, but later on they become susceptible. Sorry, very resistant in the young age, but very susceptible during the adult age, and it could be vice versa as well. So I guess looking at the literature and the biology of the disease is very important when you're trying to assess this in the field or in the glass house, because if you get a data on a very wrong timing on
Mhm.
in a particular stage where everything there is resistant, then you're likely to have a problem later on when you plant these materials by, like, let's say a farmer has planted it because you said it's resistant, so it might
Mm-mm.
succumb to infection later on at a particular stage. So I guess with the resistance screening, if you can do resistance screening from seedling stage to harvesting, that would be good, because you will have a data that there was no onset of disease on that particular stage. But for bacterial wilt, you can start screening from the seedling stage because the bacteria can enter the host plant. But as they mature, the plant as the plant matures, there is a tendency that they become more resistant to bacterial wilt because the roots become more vigorous. So the plants become more vigorous as well. So they fight, like it's like an immunity that they fight against the infection because of their the vigorous growth of the plant. That's why fertilization is also important when you're trying to fight against plant diseases, because the more healthy the plant is, the more likely that they can fight against the infection of these diseases.
Okay, that's great. So, so class, as you've seen, it's quite clear now in us that it does depend on the on the pathogen, or and of course the species of the crop and the environment as well. So, sorry, so is it possible that if we have that you could have low test or pathogen density and you can't do anything about it in a particular season, and then test in another season with high pathogen density and show different rates of resistance within the same genotype?
Yes, that would be something to consider when you're comparing different environmental conditions and different inoculum pressure as well. That is why, um, again, you go back to the biology of your pathogen, which is the best time to plant, which is the best area to plant. So that's why you call it a nursery area, because you know that there's an abundance of inoculum. But at the same time, you need to know when is the best time to grow your plant as well. So if your crop is not during the seasonal months, you're likely to stress your plants as well, and you're likely make a very, let's say, all of them could be very susceptible because they're stressed. They're not in their season. At the same time, if they're not in their season, it could be that the pathogen also may not be able to cause the disease. So, for trials that involve seasons, yes, it's always good to know where to plant and when to plant your test materials. But if you're doing glasshouse trial and you have a condition that would create an environment favorable for plant and favorable for the pathogen, then that's okay. That's why there's what we call speed breeding. You can do this particular activity in the glasshouse, even if it's not the season of that particular crop. So it helps. That's why we do glasshouse trials, because it we create an ideal environment for the plant and the bacteria or the fungi without considering the effect of the environment. But if you're doing environmental assays or environmental trials, you are likely to get differences between season one and season two. But what you're going to check there is the pattern. That's why you need to have a control for trial ones, control for trial two. And you just need to check. You will not get the same values for trial one and trial two, but if it's really resistant or susceptible, you will have the same pattern. The ones I show you the graph. So maybe in season one, your resistant check is, let's say, 0, and your susceptible check is 15. In trial 2, because it's very it's a season and it's very ideal for disease, you may get that your resistant will have 1 and your susceptible will have 30. So there's that 15 increase, but don't look at the value. You look at the pattern, that there is a doubling of the values from trial 1 and trial 2, and you will see that those resistant lines are between those resistant and susceptible. So it's not always that you get the same results in trial 1 and trial 2 for, or season 1 season 2 in the field, but in the laboratory, it's most of the time it's always very within that range, because it's a condition that is repeated at regardless of the months, regardless of the year. It's the same environmental condition. So yes, environment plays a big role in data gathering, in the disease expression, as well as it could affect, yes, it could affect the screening procedure. But yes, look at the pattern, not just the values of your data, but look at the pattern from one season to another. If you're not happy, then you do another test. So at least 2 trials and then probably additional 2 trials, one each for every season, wet and dry season.
Thank you very much, Doc Mark. So I think a very well seasoned experience with regards to these screening procedures has really optimized a lot of these processes, particularly for anthracnose and bacterial wilt and plant pathology, IPB. So any questions from the students?
Uh,
Please, Kate,
so
go ahead.
one question, sir.
Mhm.
Sir, like we have studied that the new varieties which release or produce are the advantages. One of the advantages is that they are more resistant towards the new diseases, and they are more stable and they are more productive. So it's a basic theory in the production of the breeding, plant breeding. But the history suggested that like most of our earlier plants and earlier varieties, they do not have any, like they are more they are more resistant towards the diseases
Mhm.
and than the current one. So my question is that, are we like degrading the quality of the plant, or is it that the pathogens are developing?
Mm-hmm. Well, that's a very good another and a very good comment on our question again, because the reason why we do plant breeding every year is because either there's an emerging pathogen or there is a new strain of that pathogen. So with plant breeding, we always have to consider that we always want the commercial trait. So if it's high yielding, then you need to incorporate high yielding with resistance. Now, when your plant is planted in an area with a new strain, you are likely to get a susceptible variety or susceptible plant. That's why with in IPB, we're doing it in a holistic way where we need to monitor which area has a strain A, strain B, and which region has a more destructive region. So if you want to incorporate resistance of every strains you have from different regions, then you need to incorporate that in your pre-breeding activities to make sure that when you deploy the resistant lines, they are considered as resistant across different strains or different species. But again, that's very difficult because when we develop lines, it might take 3 to 5 years, like for a tomato to be developed. And by that time, you will have a new strain of the pathogen because of the constant change of climate, the weather. They also evolve with the environment and their plant as well. So if your resistant line also is, let's just say, stable only for different a particular group of pathogen, and here comes another group of pathogen, they may collapse, they may break their resistance because they can't fight the pressure. And that's so they're still resistant to this particular strains, but with new emerging pathogens, they become they break their resistance. So that's why there are methods now that are being developed to speed up the process of the breeding, because if it takes 5 or 10 years to develop one variety, and after that 5 to 10 years you have a new strain more destructive than the previous one, then that's likely to cause a problem. And so we always, and you're right, Sajay, we always screen that with the really recent pathogens or diseases, and not the previous strains that are only, let's just say, they were cultured and collected and stayed in a culture collection. So we need to go to the field and collect actual pathogens that cause the disease. So it has to be real time, within probably within 3 years those diseases have occurred, but not maybe not after before 10 years, to make sure that you're using the most updated pathogens that are in the field.
Yes, that is right. Thank you very much, Dr. Mark, because I think these pathogens are constantly evolving as well and poses evolutionary risks to our resistance varieties that have been
Mhm.
registered. So we need to constantly keep up with the arms race. So any more questions from the class? Go ahead.
Very quiet. Parang first, may Starbucks ata si Sir Rio dun sa mga nagtanong.
Yes. So any questions? Yes. How about let's try to ask some person. How about Sue? Do you have any questions? Sue is from. Let's try to take a look if students from other parts of the world did have any questions. Sue, are you there? She's from Myanmar.
Yes,
Any
sir.
questions? You have any questions?
No, yes, sir. I'm not familiar with this topic. That is why I am think about, sir. Thank you for the nice presentation, sir, and the discussion.
Mhm.
Thank you.
Okay. So they are probably bombarded with all of the new information about plant pathology and disease resistance screening. And how about we ask? Let me check. There are a lot more people present. How about uh, Alexandria? Are you there? I think yes. She's from UAE, or if I'm not mistaken, Alexandria or Marajun.
Belgium.
Marajun, are you there?
America.
Yes, sir, I'm here.
Yes. Do you have any questions?
Uh, no, sir, I was thinking about the last question of Saita and regarding the disease resistance, degradation of disease resistance of the current varieties. If I
Mm-hmm.
find any question, I will ask, sir.
Okay. Okay.
Okay, so thank you, sir. Good afternoon.
Okay. So no questions so far. Going once, twice.
Sir,
Yes.
one follow-up question in my previous question, sir.
Mm-hmm.
Like the previous question, we have been developing many resistant variety. We have been doing many, like we have developed many technologies and many advantages. So like it's just a normal question. Like, are we like achieving the goals of as a plant breeding? Like, are we going to the, like, I mean, in the right side, or is it we are going in the left side, sir? I mean, the left side, the sides which I'm talking about, it's the, like, we are advancing or we are lagging behind. Like, as the history suggested that our previous, our ancestral plants, they like, although there have been some infestation of diseases and pests, like they can withstand like many diseases, and or those who haven't been able to withstand this, they have been selected by the nature and removed, and they have been evolved into other varieties. But like normally they have, like they are more, I mean, my general point of view is that they are more what to say, advantageous. So like we as a, like, not we, like, I could ask you, sir, like, are you that can you say that you are advancing towards the betterment, or are you like lagging behind? Like, as the once the technologies and all this advancement have been arrived,
Okay.
like we are facing more and more problems rather than providing the answers.
All right. Yeah. Thank you, Satya. So I'm put in a situation where I have to put opinion and science together. Um, but I've from what I've always heard is that there's there's no such thing as
Right, sir. Your internet connection, sir, I think got cut off.
there's no such opinion in science, because what you see in science is what you get. From a science perspective, I think we're really moving forward. There's more methods, there is this difficulty of there's a lot of companies. Ah, sir, okay na po ba, sir? Sir Rio, is it much better? Okay, good, good. Yun na pa lang yung orientation ng mobile data. Anyway, so going back sa question is Satya, I think it's from a science perspective, we've obviously moved forward and have advanced with the procedures like speed breeding. We have procedures in plant pathology to expedite the process of screening resistance. And we have a lot of resistant varieties that have been produced by private companies nowadays. I think the problem, not problem, and this is just an opinion from an observation, is that the adoption of these materials do not go well with the commercialization of this material. So not everyone are able to afford materials that are considered resistant or high yielding because of the prices of these seeds that are being commercialized. So I think we've obviously moved forward. We've obviously solved problems, prior problems, but then, like mentioned earlier, while we're solving problem A, there's this problem B that will potentially occur in the next few years as well. So there's this continuous solving of problems, and I guess for a farmer, it's very difficult to choose which variety you're going to plant, because you always have to consider how much do I have to pay for that. So and also with the question if we are really moving forward and solving these problems about diseases, it's really from a different background or different perspective. But if you're asking me from the perspective of a plant pathologist, I think we've done a lot of work that has advanced our research and our effort to solve problems. And from conventional works, we dealt with now with molecular works to expedite the process of identifying novel lines with novel resistance to pathogens. So I don't know where you're coming from, Satya. Maybe you're looking at the economic perspective as well, because probably that's an area where there's a bit of a lag behind with the adaptation of the technologies and adaptation of the commercialized varieties of these plants and vegetables or fruits. But there's too many materials that are being commercialized every year. But the question is, how many are being adopted by farmers? And I think that's why you see the gap there, that not everything that is being produced are being adopted by farmers or growers. But we're obviously advancing, I think, from my perspective. But yes, I don't know with others if they would agree with that, but yes.
Yes, of course. And probably also, Satya, you're probably thinking about the part on the agroecological. If it's on the agroecological perspective, more and more varieties are not just only bred for conventional means, but also for organic means, and as well as those varieties that could tolerate various ecosystems that are niche and could be different from one microenvironment to another. So in terms of expressing their resistance. And so, I mean, there's a lot of research ongoing and there's still a lot to be known, but it's a really interesting field. But going back, sir, as you said about molecular markers, about doing breeding, could you help our students also try to understand any studies on molecular markers in speeding up the breeding process, sir?
Okay, thanks, Saria. The molecular markers are used to identify which lines are that are potentially resistant before you test them in the field. So, and these markers are more useful if you have a perennial crop like fruit crops. So instead of waiting for 10 years for a coconut to show resistance, with the use of molecular markers, you would be able to identify which particular plant carries the gene for resistance. So instead of planting them, waiting for 10 years to show if it's resistance or not, um, with the use of molecular marker, you will be able to identify this particular plant. However, you still need phenotyping. So you can only confirm that until you see that this particular plant is um, resistant. But if you're doing breeding and you have a lot of these F1s, F2s until F6 or S6, molecular markers are very, very, very useful because you will be able to shorten the time to select for these particular materials without phenotyping them. You may opt out of phenotyping, but towards the end you still need to phenotype or to assess the resistance against the diseases or pathogens that these materials are supposed to be resistant of. So the aim of the molecular marker is really just to expedite and to shorten the time of screening procedures for identifying the resistant or susceptible lines. But it's not just as simple as molecular marker, because sometimes it could be complicated because it might not be just one marker, but it could be multiple markers. So there's always a science thing that is still complicates me. I think Sir Rio is more knowledgeable on that than me, but it's fascinating. It's really nice if you do molecular mark. One of our projects in bacterial wilt, we look at different molecular markers, and we found out that some of these tomato lines that are resistant in other countries are susceptible in the Philippines, and that is because there are still molecular markers that needs to be um, explored or identified. And that's part of the research that we're doing. What other markers that confer resistance that would allow us to identify materials that are considered as resistant, not just in other countries, but also here in the Philippines. So yes, I think in iCrops, if you're working with plant breeders, you are likely to do some molecular marker identification, or maybe you use molecular marker to find a particular trait of interest. Yeah.
Okay, that's great, sir. Thank you very much for sharing with us that information. I think some students here are also ABT students, so they might be more fascinated on the molecular side of things, on the biotechnological side of breeding. So, any more questions from the class? No more questions? Are you sure? None?
Yay!
Okay.
No more
So,
questions.
if
I
there
have a
are,
question.
there is one
Okay,
question
go
from
ahead.
Georgia. Go ahead.
So it's not a much important question. Actually, I'm gonna ask about the resistance summer variety of tomato, resistance against bacterial wilt. Actually, sir, I am working here in a agriculture farm as an intern agriculturist, and every year we need to do grafting of rootstock and the
Mhm.
summer variety of the tomato for preventing the bacterial wilt. And that's why
Mhm.
I'm asking that is there any release summer variety of tomato which is resistance to bacterial wilt in Filipino varieties, sir? I'm asking
Mhm.
about.
Okay. In the Philippines, we don't have, let's say, summer variety. We don't go to the supermarket and ask what summer varieties do you have. So usually, I'm not going to mention the variety name and company as much as I can. So, but these varieties, as we go when we go to the field, um, they're not very susceptible, but they can still get infection. And we haven't had any experience where we saw a farmer doing grafting or using rootstock as a method. But the thing about rootstock method is that it's laborious because you need to cut the upper part and then replace that with a what you said summer crops that are resistant. But if you're doing rootstock method, here in the Philippines, they recommend using eggplant varieties that are resistant to bacterial wilt because there are more resistance to in eggplant than in tomato, and then use tomato as their scion. So the rootstock is eggplant, and the tomato is the ones in the upper part. But with regards to varieties, I'm not very aware of any variety that you would say resistant to bacterial wilt, because a lot of these varieties I've seen in the field are susceptible to bacterial wilt. Even with the mostly or the popular varieties that are mentioned by farmers, they all succumb to infection when we went to the field, especially when the area is like after a rain, you're likely to get after 1 week some infection. So that's the reality. It's very difficult to find what we call really resistant cultivars here in the Philippines. So you need to add other control measures if you'd like to. But grafting or the rootstock method is very useful, I think, if you're doing some greenhouse or um, a small field, small area planting, because it
Sir,
could be very
yes,
laborious.
yes, sir. Yes, grafting is useful, but sir, I was thinking that as a farm like our that supply the grafted tomato plant, but if there
Mm.
is any resistance variety, genetically resistant variety, then it will benefited the farmer directly, I think, because then
Yes,
there
that's right.
will no business another business, and that's why, sir, as I am an agriculturist, that's why I
Yep.
just thought about that, that if we can develop any resistant variety then it will we can supply it to our farmer directly.
Yep, that's
Yeah.
right, Mary June. And I hope that in the coming years, these varieties would be available to anyone who are doing business, particularly like you and your farms there. We hope that they will be available to everyone who are expected to use these materials. And that's part of the project that we have. That's why we partner with World Vegetable Center, because these materials are likely to be released across Asia and probably in the world later on. So yes, we are all hoping for the best, and I hope that one of the products of our research will be useful to you in your farm there. I hope so. I might be the researcher who started it, and someone might, after 5 or 10 years, someone might do another research continue that will continue my research. So I would be happy to see in the future that your farm is using or will be using one of our products of our research here at the Institute of Plant Breeding.
Yes, sir. Thank
Great.
you so much, sir.
Thank you very much for that wonderful question, Arjun, and of course, Sir Mark. Yes, I think hopefully that is one of the end goals of the research. Of course, is to be utilized by the respective stakeholders. And that creates a big impact, and as well as also a sense of pride, as well as for researchers and as well as honor, of course, to have been able to touch lives of different people through their research outputs. Now, any questions so far, aside from the group on screening and of course breeding for assistance? Any more? Are you sure? Doc Mark is a busy person, and I know that it will be a very rare opportunity that we get to ask him again. But although you can reach him through various channels, including their respective web page, as well as Twitter account and Facebook account. Dr. Mark is really quite uh, really active on those specific social media platforms. So you can more likely reach him from there if ever you have questions. So if none, then let me present this.
Sir,
Yes, yes.
one last one. One
Go
last
ahead.
question, sir.
Yes.
Okay.
Like this concerned me that like we have been developing many resistant variety. Either it can be a GMOs or just a local conventional method. But my concern is that my query is that how much, like let's take an example for the GMOs. Like by how much degree we can like change. Actually, we are changing the genome of an organism, like if we are to develop a resistant variety. So like by how much degree can we actually alter from a particular variety, so that we can say that it's the variety which has developed. But like my main concern is that by how much degree can we change the genome of an organism or of an variety, like, and not only about the GMOs but for also the conventional methods, like how much is it that, is there any criteria, or is there any government, I mean bodies that administer such topics?
All right. So, well, thanks for that, Satya. I think regardless if you use conventional or genetically modified, there will always be a change in because they always naturally cross. And the thing with genetic engineering, it's more precise than the crossing that happens in nature, because everything is they genetically change. So by how much is a question that I could not provide you an answer, because probably I'm more of a geneticist, and a plant breeder would be able to provide you a precise answer to that. But um, as you've said, there's always going to be a change. And the thing about genetic engineering is that it's more precise than the conventional, because with genetic engineering, you're changing just a particular section of the DNA sequence. And that is why um, some argue that it's not good to change that, because you're changing it, but in natural condition you're actually changing the genome of a particular plant or plants, because they're crossing in nature. So by how much is something I could not answer at the moment, but it really depends, I think, on your crop, because there are crops that might require more time or years before they show the change that you expect them to see. For instance, the change in the leaf size, the change in the fruit size. um, I don't know. I would be 100 years old to see that the mango carava would change into a different shape. So we don't really know. So I guess the more sexually propagated the plant, the higher the rate of change. The asexual ones would probably remain as it is, because it's cloned from a uh, a stem or a flower, or grafted. But yes, I think it depends on the biology of the crop as well. So uh, that would be my last piece of advice for that one. But yes, it's a constant change. But by how much and how long is something, it depends on the crops that you're studying.
Yes, and do take note that the technologies with regards to these fields is advancing in an exponential rate, in such a way that even right now they are talking about plant artificial chromosomes, synthesizing artificial chromosomes. And so with that, you could incorporate a lot of resistance genes and those artificial chromosomes back to plants. So some of these it's advancing in a rate in such a way that policies could not keep up. So some of the policies are still not yet in place. So even when CRISPR had started, some have had a debate about it, if whether or not, if it's just when you knock down a particular gene, would it entail genetic modification, or would you need biosafety levels, and all sorts of questions that come along with the technology as well. So this is still again a developing field, and a science has grown so exponentially that policies and even thinking about it, whether if it's right or wrong, and the ethics and the consequences are still yet unknown for some even. Okay, so probably with regards to that, there's no hard fast rule. That is the definite answer, probably for now.
Yes, sir. Like that was my question, sir. Not I'm not asking about the actual degree, but like
Mhm.
we could like attain a physical form, but we could change the entire internal structure. So like who is controlling them and who is governing them? That was my question.
Yes, yes, yes. So right now you can even do it until in a chromosome level. Synthetic chromosomes are a thing nowadays. So I don't know if hopefully a synthetic genome, but so far right now they have not been able to produce a plant based solely from genetic sequences. So that's one thing that's different. So they need some sort of a living cell in order for it to be a host that would be incorporated with a specific vector. So there's a lot of things going on in that field, and probably many would not agree, particularly some agroecologists, and it would depend on which perspectives you choose. But try to be as logical and, of course, be scientific about it when you deal with these things. Okay, so that's a good question anyway. So, any more questions? None. Then I will present maybe the certificate for Prof. Mark for graciously sharing with us. I will just read the citation. Institute of Crop Science, a certificate of appreciation is presented to Dr. Mark Angelo O. Balendres for his valuable contribution as resource speaker to the Agri 150 Methods in Plant Breeding 1 laboratory class, which is held with his lecture titled General Methods and Practices in Biotic Stress Resistance Screening, held on the 12th day of May, given the 12th day of May 2021, University of the Philippines, Los Baños. Signed, yours truly, and of course, our Deputy Director for Coordinating for Instructions. We're still waiting for the signature, Sir Mark, but definitely I will send the certificate once it's signed. It's still in the email, yeah. But I have already updated Dr. Edanio. Thank you very much, sir. This has been a wonderful learning opportunity for everyone, and I hope the students coming from various backgrounds, various classes, but various provinces had learned a lot from you today. So, in behalf of the class and the teachers and the lecturers, and of course, my partner laboratory instructor, Prof. Leslie, again, thank you for gracing us this afternoon. We are in debt. And with that, I think we end our laboratory session for this afternoon, and I hope to see you next meeting. Thank you very much. Maraming salamat po.
Thank you very much, everyone.
Thank you,
Thank you,
sir.
sir.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you very much.